Okay, hello everyone. I was going to try to get. I started working at the Neurosim lab with Bill before I started working with Salvador and developing NetPyNE. And when I was working with Bill, we were doing this embedded ensemble in coding theory, exploring some ideas about how the cortex works. And yeah, NetPyNE didn't have. I mean, it could run batches, but then in order to make plots out of that, that didn't really exist much yet. And so I had to sort of develop my own code to display the analysis from batches. This is very similar to the way NetPyNE evolved. We run large-scale, multi-scale models, and we need a way to set them up. And so NetPyNE evolved as an internal tool to do that. And a lot of the additions to NetPyNE evolve as internal tools that eventually we generalize and make accessible in NetPyNE. Once I'm done getting sort of plotting and analysis in better shape, then the next big push will be for batches, so that we can make run batches very easily and then analyze them and have, we looked at the plots yesterday. There's lots of great plots for individual simulations. But if you're running multiple simulations right now, for the most part, you have to do your own analysis to pull that all into place. So if you plot traces and you want to compare them, you got to, you'll have, if you have two parameters with 3 values each, you're going to have 9 simulation outputs that you want to compare. So you want to pull the data from each simulation and plot it, and then put it into a grid, which is basically what this is here. So yeah, so the code I had that worked 2 years ago, but now it does not work anymore. This would be a 1, 2, 3, 4, 5, a 5 by 5 batch. So basically what I did was I varied 2 parameters in the model. And for each of those 2 parameters, I gave it 5 values to explore. And so 2 parameters, 5 values each means 25 simulations this had to run in order to get data out for these figures. Okay. And yeah, so really when you run a batch, so you got one parameter, which is the NMDA rise time. Whatever, you got one parameter on this varying along this way from 1 to 5 to 15 to 30 to 60. And the other parameter is glutamate stim from 0.01 to 0.012 to 0.014. Okay, so you got these 2 parameters set in space. And usually what I do is the default values are going to be the center of my exploration. I usually explore in both directions, right? So this center square would be the default values, an NMDA rise time of 15 and a glutamate stim amplitude of 0.012. And then we want to know what goes on if we modify these parameters a little bit. So I'll expand it up, upwards and downwards in value in both parameters. And you'll end up with a nice parameter space, a grid search. This is not evolutionary. This is not optimization yet. This is exploration. But yeah, we started from one case and we explored varying 2 parameters in 2 different directions. And really, when you run a batch and you explore 2 parameters, this is exactly the figure you want output immediately. You don't want to have it spit out 25 figures and then arrange them in space and then look at them. You really want it to be automatic, and that's our goal. And we will get this code into NetPyNE over the next year or so, sooner, hopefully. Yeah, so let me, this is what this weekend I will, it used to work with NetPyNE, but NetPyNE has evolved in the last 2 years, and I haven't kept up with this work. So I need to adapt this so it works with NetPyNE as it is today. And then next week, I will share this code with you. And when you run batches, you get 3 by 3 or 5 by 5, whatever. It only works for 2D comparisons right now. We're going to want to expand that into 3D and 4D and 5D and stuff. But right now, you can only look at one parameter, and compare them across 2 parameter space. And yeah, so yeah, you want to explore your parameter space. You want to make sure if you vary a parameter a little bit, it doesn't explode. You want to make sure your model is robust to small changes in parameter values. If you change a parameter by a tiny amount and your model explodes, then it's not a robust model. Yeah. So at this point, I think we've got some time because I didn't get my work done in time. Let's go through. I'm going to talk about how I got involved with NetPyNE and the work I did leading up to joining NetPyNE and so on. Okay. So Bill hired me to work on a project called Embedded Ensemble Encoding. And yeah, let's zoom in here. Yeah, just to put some names here, me and Salvador, Sergio, who's an MD PhD, now in his MD phase, Sam Nemotin. Surgeon is our experimental collaborator. It's great to have an experimentalist you can work with. And of course, Bill worked on this also. And so, yeah, so we were looking at dendritic plateau potentials. So this is a zoom in on the soma and basal dendrites of like a pyramidal cell. And one particular dendrite we're looking at here. And what our experimentalist does is he does glutamate uncaging experiments combined with the voltage, the calcium potentials will create light depending on how much calcium concentration there is. I forget the term for that. If somebody knows it, spit it out. But yeah, essentially he's uncaging glutamate. Glutamate is the most common excitatory neurotransmitter in the nervous system, and it's used by many, many cells. And normally synapse releases just a little bit of glutamate to tell the next cell what's going on. But there's a lot of interest in a lot of connections coming into dendrites tend to cluster, especially if they're functionally related. So you get a whole bunch of synapses coming onto one small dendrite or one small section of dendrites. And if you get a big glutamate burst, like if a bunch of those cells all fired at once, you will actually activate the dendrite. So in the past, dendrites were often seen as sort of passive cabling. You got an input and it's just the dendrite is just the cabling to connect it to the soma. Now we know that's not true. Dendrites are active and they do a lot of processing themselves. And you can actually get two states in the dendrites of pyramidal cells in the cortex. So you got a down state where basically they're quite a ways from their spiking threshold. It would take a lot of activation to get them to start spiking. And a lot of cells will sit in this down state and normally. But you can bring cells into an up state. We call it an up state. If you get a cluster, a ton of glutamatergic input in one dendrite, that dendrite will actually cause a plateau in the dendrite. So the voltage will rise to a new level, which is regenerative. So it'll stay at that level for a period of time. And of course, that excess voltage in a dendrite is going to flow into the soma. And you can get a plateau in the soma as well, which means that instead of sitting at a very low voltage, now all of a sudden it's sitting at a voltage very close to its threshold. Okay, so when you're in the off state, it's going to take a lot to get a cell firing, but if you're in the on state, the slightest inputs will get it going. So if you can get a neuron into this on state, then it's going to synchronize with whatever inputs are coming into it. And there's a lot of interesting topics to discuss here, like the binding problem. How do we combine different sensory information into one experience? Things like this. Yeah, so we had an experimentalist. So this is largely a single cell until we built it up into a network model. You have to start with a single cell and make sure the model works at the cellular level before it makes any sense to bring that into the network level. Because if your cellular level is wrong, having 1,000 of those is not going to be any better. So one of the early steps in building up a model is making sure that your individual cells are good before combining them into networks. So again, if you activate basal dendrites, you can drive them into a plateau state, which can also drive the soma into a plateau state, in which state it's very easy to get them to synchronize with whatever inputs are coming in. And so, yeah, the essence of embedded ensemble encoding theory was that you've got all the neurons, but then you've got ensembles that, because of the inputs coming in, have been put into the active state. If you saw a lion, for example, your networks to start running would be brought up into this plateau state. So you're ready to start as soon as possible. You're ready to start going. And yeah, it's sort of an all or nothing thing. It's one state or the other. And so, yeah, so Surgeon Antic at UConn, University of Connecticut, is an experimentalist who uses voltage and ion-sensitive dye imaging. That was the word I was looking for. And at the same time, he does a whole cell patch onto those neurons, and he can uncage glutamate wherever he wants experimentally. And so, yeah, earlier we talked about you start a model and you want to validate it before you start doing anything interesting. You want to make sure it kind of represents what you're working with. And so in this case, we validated it using, in one case, back propagating action potentials. So if you get, if you got enough action going on to cause a spike in the soma, that doesn't just travel down the axon. That electrical pulse also back propagates into the dendrites. And this probably plays a large role in plasticity and some other things. But this back propagating action potential is important. So we wanted to make sure that we had the back propagating experimental data. So hey, let's use our model and make sure that our back propagation into the dendrites is pretty equivalent to what we saw experimentally. And yeah, so we talked about perturbation studies, right? So you got a model and you got an experiment. Often what you want to do is perturb it a little bit, do something to it, kick it, and see what happens. So in this case, we used, our surgeon used 2 perturbations in his experimental model. One was he put in a sodium channel blocker. So like tetrodotoxin, TTX from the blowfish and a couple other places, causes, it blocks the sodium channels. So the sodium channels will no longer spike. And obviously that's not good for life, but it's an interesting perturbation that you can explore in your model. So if we do a sodium block, and I'm colorblind, so I have a hard time. I'm going to zoom in a little further. There we go. These are 2 very similar colors. Is this green? One? Is this the sodium block or the potassium block up here?
The red one, the one right on top, that's the potassium block.
Thank you. No, that makes perfect sense.
Control, and the one at the lower is sodium block.
Perfect. Thank you.
Yeah, the potassium is always on the top, and the sodium is on the bottom. Yeah.
Yeah, which makes sense, because if we're blocking sodium, that's going to block the action potential amplitude. It's going to be lower. So it makes perfect sense that this is the sodium block. The potassium block is going to stop, after the sodium has caused it to spike. If you block potassium, it's not going to come down nearly as quickly as it does. And so, yeah, we got experimental data, and we modeled that pretty well in a fairly simple model. And yeah, so we sort of validated our model a bit there. Validated. Okay. And then once we've got a model that's validated, we like it for what we want it to do, then we want to explore it. Okay. And yes, so 2 things we explored. NMDA. NMDA receptors are what accept the glutamate neurotransmitter. So if we play around with the time constants of that receptor, we can explore the model. So let's zoom in even more here. Great. So on the top, we see the NMDA rise time constant. And on this axis, we see the glutamate stimulation amplitude. All right, so this is the lowest glutamate stimulation on the bottom, or on the top. And on the bottom is the highest glutamate concentration, the puff of glutamate that we release, a simulated puff of glutamate we release. Obviously, increasing the amount of neurotransmitter you release, moving down that chart, you're going to cause an increasing response. Turns out that slowing down the NMDA rise time also causes a larger response. So here you can see it rises very quickly. It starts to rise slower and slower and slower. Slower rise time means it's active for longer. And yeah, so this was a nice way to start tuning our model against 2 parameters to see what we wanted to do. And again, this I ran a batch, a 2D 5x5 batch simulation with the NMDA decay, in this case 5 values, and the glut stim 5 values, and that spits out 25 sets of data from simulations. And then your analysis code wants to be able to import all those sets of data, arrange them according to your 2 parameters, and make a nice plot that's quick and easy. And so next week, you guys will be able to use this for batch simulations. You could do a 2x2 batch, 5x5, 3x3, whatever, and it'll plot your traces or whatever figure you're interested in a nice grid like this. Yeah, so we were interested in spines for a period of time. We talked a bit about spines. A lot of inputs into excitatory neurons in the cortex come on to spines. A lot of models just ignore them, but try to compensate mathematically a little bit for them. But yeah, here we looked at the neck resistance of all the little spines. We put hundreds of spines onto this 1-cell model. And the same thing, the glutamate stimulation. So just increasing stimulation as we go down, and increasing neck resistance as we go up. Experimentally, we saw something kind of like this. So that fit within our range of neck resistances that would be expected. So this, this is a way to sort of choose your values or hone in on values for your parameters. This is what we saw experimentally. So let's make the neck resistance this value. Spine distribution. Yeah, we distributed spines in different ways. And yeah, so we compared our model with the experiment. This is experimental data with increasing amplitudes of glutamate puffs as you go up this way. So this is very little neurotransmitter. This is a lot of neurotransmitter. And we modeled a glutamate puff. You have to get kind of fancy with modeling experimental perturbations in your model, because I could model a glutamate puff like this with RxD, but we're less interested in sort of subcellular responses as we are in cellular and network responses. So we don't really want to go down to that low of a level. So the way we simulated glutamate puffs was in our net params. You can set like a location or a dendritic branch you want to do a puff on, and you set the middle of the puff location. And then what the model does is, in the middle of the puff location, it adds a lot of neurotransmitter. And as you move away from the middle of that puff location, it gets less and less glutamate hitting the synapses there. You know what I mean? So it's like we're not really modeling a puff of glutamate. We're saying that were you to put a puff in right here, we would see the most neurotransmitter hit in the middle, and then it would decay spatially to both sides. And we played around with spatial decay values and temporal decay values to get something that sort of matched experiments. But then, yeah, we've modeled a perturbation that you can put into this. And in our model, when we increased the glutamate stimulation, we got it to reproduce the experimental results quite well. And then, yeah, so this was early on when we had just sort of started really validating the individual cell model, but we wanted to start including it in network models right away, because these, whenever a bunch of synapses came into one small area, we could simulate a glutamate puff and see, hey, how does the network respond when one of the cells in it gets a dendritic plateau potential and a somatic plateau potential? When a cell is in the on state instead of the off state, how does that affect network behavior? And at that point, you want to put a bunch of these cells into a model and then start exploring the raster plots and all of that stuff. We're at 3:40. Yeah, let's top questions, discussion. We could talk for a while. Otherwise, we're just going to end quite early. Any questions or comments? I got to give you guys a little time to think here.
Hey, Joe.
Yeah.
Hey, Joe.
Yeah.
I have a question.
Yeah, please.
So in this batch for the optimization and exploration of the parameters, can this also be applied to the reaction diffusion thing? So like exploring different parameters for diffusion coefficient or tortuosity or everything else. It's also okay.
Yeah, this is very generalized. So any parameter in your model can be pulled into the batch, and you can explore that parameter.
Okay.
Sometimes it takes a little doing, because what you have to do is you have to have it as a member of your simconfig object, because batches only change numbers in the simconfig object. And so, yeah, so you would say simconfig dot sodium channel density equals this, okay? 0, 1, 2, whatever value it is. You could set it to whatever it is in your simconfig file. And then in your netparams file, you have to connect that value to your model. So if you want a very sodium density, then in your netparams, you'll have to say, you know, for whichever neurons you're looking at, for each seg in this neuron, set the sodium channel density to this sim.config sodium density value. Okay, so anything you put in simconfig, you can vary in batches, but you have to connect that to your actual model in netparams. Sometimes it's direct, you know, sometimes it's there's no transformation required, like a lot of the channel parameters and things. You can just set that directly. But for a lot of things, you're going to have to connect it in netparams to affect your model as that parameter you're exploring as its value.
And this will, sorry.
I was going to say that we have an example showing parameter optimization that links RxD to a network, and we just finished making that work last night. And I thought I was out on another meeting when Craig gave a talk, but I thought he was going to include it. So maybe if it's still working, which I think it is because it was working last night, Craig can maybe show it, or I can show it in a bit. And that's a nice example because we modified parameters. Sorry?
I'm planning to show it a little bit in my batch sims as well. So
awesome.
Yeah, it's
from RxD to the whole network?
No. Yeah, just like how you can export a parameter from the netparams file into the config and then change that in the batch. I'm gonna show that a little.
Oh, great. Yeah.
Yeah, Salva, the RxD one, I didn't actually like optimize a parameter, just ran batch simulations over a parameter space.
Yeah, but I think that's interesting. If you don't mind maybe showing that, Craig, because it links all the scales, even if it's not like a very functional thing.
Yeah. No, the collab is in the chat right now, and I think I posted it on the Slack as well.
Okay, great.
Under Ask Tutors, right? I think.
Okay. You wanna maybe go over that? Like.
Oh, I mean, well, I can go over it in more detail, but I did go over it during the.
Okay. Sorry. Okay. I was in another meeting, so I missed that. Okay.
I don't want it.
I was busy trying to get my batch code working, so I wasn't paying attention to Craig either.
Jesus, guys.
Nice. And so the other thing I wanted to show quickly is I forgot to show before some of the code I have for analysis in the.
You're muted, Salva.
sorry. So I will quickly just show that. It's in line with what Joe was talking about. Window here. So if you see my screen now. So this is the tutorial I was showing before, the NetPyNE batch 8 tutorial. And we got up to this point where we run these 9 simulations, 3x3 parameter exploration. And I showed some of the results, which were actually here in the folder, in the output folder. This, for example, the raster. You could see one of the rasters here. But a nice way of visualizing this kind of batch exploration is to look at all the parameters at the same time. So in the cell below, there is this code called analysis.py. And it's just a function first to read the data. So it just reads the data from all the different output files in JSON. And it then sort of combines the data into a single data structure and generates a plot. So it uses pandas, which is this Python tool, which is very useful for analyzing data. So if we run this code, should produce a nice plot. Here it is. Okay, so if you run it, you will see here is in the colors represent the firing rate of the M population, which is the one we were sort of looking into. And in the two axes, you have the decay constant of the synaptic mechanism, the 3 values we explored, and the x-axis, the 3 values of the connectivity weight. So you can see that it's quite an interesting result here because it's not very linear at all. So you see the two extreme weights kind of produce the lowest results. Well, not really even. So the highest firing rate is when you have the connectivity weight of 0.01, which is likely because lower weights don't reach the threshold for spiking, and this higher weight might be too much. And so overall, you can see the relationship of this firing rate to these two parameters. And you can use this code and adapt it to some of the analysis that you might do. So yeah, so I just wanted to show that example. And if there's any questions now for me or Joe.
And obviously, batches are very important, but we're just sort of starting to get to really making them robust and really solid in NetPyNE. So if you guys are interested in helping us do that, you know, we're always happy to have collaborators.
Yeah, we actually have tons of code to analyze batches from both the M1 and the A1 models, and we're just working our way into adapting it to make it generalized so it can be incorporated into NetPyNE. Any questions? Okay, so I think we can resume in 10 minutes. We can start this last session, the exercises that Joao is gonna go over at 4 p.m. and then we can finish at 6, and we can all have an early release on Friday. And so I won't see you until Monday. Hope you have a great weekend. And please send me by chat by tomorrow the name of the tentative title of your project and the names of the participants. And over the weekend, we will make sure to assign appropriate tutors for each group based also on your preferences and the topics of interest of each one. And on Monday, Joe will give in the morning a session on how to install all these tools on your laptop, both for Mac, and we will try to figure out for Windows as well. And then remember, we will have invited speakers every day. We have 2 invited speakers every day, which will tell us about some of their modeling work in NetPyNE. So that should be quite interesting and probably give you some ideas as well for your projects. If you have any questions, please contact us on the chat. And well, thanks everyone for attending the first week, very intensive week. Hope it was useful. And I'll see you guys next week.
See you, Salva.
See you.
Yeah, so 10-minute break, and we'll be back at 4.
Thank you.
Thank you.
Thank you.
Thank you.